Hi class, we're going to dive into chapter 16, which is all about metabolism and energetics. Last chapter we were looking at the digestive system and how all of the nutrients came into the body, and this chapter we are going to be looking at how our body uses those nutrients that we just brought in. It's all about metabolism and energetics. And this chapter can be a little overwhelming. It's kind of like the biggest taste of chemistry you've had since chapter 2, I think. So just hang in there. You'll survive. You'll get through it. Let's define some terms. First of all, metabolism. Metabolism is all of the chemical reactions in a organism. It's composed of two types. There's the breaking down reactions, which are the catabolic reactions, your catabolism, and the anabolic reactions, or the anabolism. This is the making. So you have the breaking and the making, and those make up all of the chemical reactions, which is your metabolism. So your cellular metabolism is all the chemical reactions within cells. The metabolic turnover is the process of breaking down and replacing cellular organic components, except for DNA, which remains consistent. Cells obtain the building blocks from the process of catabolism, which releases energy, also the absorption of organic molecules from all the interstitial fluids. That all makes up something called the nutrient pool. So in other words, your body's taking the nutrients that you just brought in from eating, breaking them down, catabolism, storing that stuff for later, and then rebuilding it up into new parts. This is metabolic turnover, how we use and how we recycle these molecules as they come into our body. So cellular catabolism occurs in the mitochondria. This is 40% of the energy that is from our food is then captured in the molecule of ATP. The rest of it is used for heat. And that's how our body stays warm at that 98.6 degrees. Here you see an overview of the metabolism process. You have all of the organic molecules, amino acids, lipids, etc., coming into our cells. They are being broken down and, well, that's weird. They're being broken down and built back up and form something called a nutrient pool. The nutrient pool can then be used to create lots of ATP through aerobic metabolism. As that aerobic metabolism takes place, 60% of the energy from those nutrients is lost as heat. 40% of it goes into ATP. And ATP is what cells use for everything from contraction to movement to making new proteins to, active transport and all of those things. So one of the major components is called the citric acid cycle. This is where cellular ATP production begins in the cell. Organic nutrients from the nutrient pool are broken down into smaller fragments, and the fragments are absorbed by the mitochondria for processing. A substance that's used by the mitochondria is called acetyl-CoA. Acetate is attached to a coenzyme A to create acetyl-CoA. Coenzymes are basically little molecules that bind to enzymes to make them work, or to make them work better, really. They act as little hydrogen carriers, and they're usually derived from vitamins. The first step of the citric acid cycle is when coenzyme A is released and citric acid is produced. The series of steps throughout this cycle remove hydrogen atoms and transfer the hydrogen atoms to other coenzymes. Little ATP molecules are produced. There's just really one or two. And then the coenzymes that are produced deliver hydrogen atoms to the electron transport system, the ETS. The process of oxidative phosphorylation will then occur. The hydrogen atoms that were obtained through those coenzymes in the citric acid cycle will be used to generate ATP in the electron transport system and oxidative phosphorylation. Oxidation refers to that process of transferring electrons. Phosphorylation refers to attaching a phosphate onto an ADP molecule to form ATP. The series of electron transfer steps is called the electron transport system. This is 95% of the ATP in our cells comes from this process. Overall, this process requires oxygen. Oxygen is needed as the final electron acceptor in this electron transport system. If oxygen is not present, anaerobic metabolism will take place and almost none of this ATP will have been produced. So it joins with hydrogen atoms to form water as the last step. Without oxygen, the cells are going to die from lack of ATP. So let's start the whole metabolic process. Chemicals break down in the cytoplasm. Little fragments go to the mitochondria. The mitochondria combines those substances like little parts of glucose or little parts of proteins, etc., combines them with an acetate and a coenzyme A to form acetyl-CoA. Coenzyme A is released, and what we have now is citric acid, which is why it's called the citric acid cycle. And then the citric acid cycle removes a bunch of hydrogen ions to form these coenzymes. The coenzymes will produce, will carry the hydrogen molecule or hydrogen atom over to the electron transport system. And that's where oxidative phosphorylation occurs. The coenzymes bring the hydrogen atoms over and ultimately generate lots and lots of ATP and water. So briefly describe the citric acid cycle, explain the role of the ETS, and explain the link between ETS and oxygen. Those are your 3 review questions for this beginning section. Let's talk about the nutrient pool. So these are sources of substrates for catabolism and anabolism. This is the key to a cell's survival. The nutrients are used for replacing things like membranes, organelles, enzymes, structural proteins, etc. There's a constant need in the cell for ATP. So the nutrient pool provides the materials that can be broken down into those 2 carbon fragments for use in the citric acid cycle. So things like fatty acids, glucose, amino acids all make up this nutrient pool. They can be broken down into 2 or 3 carbon chains, which can be used in the citric acid cycle and electron transport system to produce lots and lots of ATP and water. Most tissue cells continually absorb and catabolize glucose. It can shift to other substances like fatty acids or amino acids in times of starvation. Neural cells need only glucose. They can only use glucose. The reserves are mobilized when the absorption of the digestive tract is insufficient to maintain nutrient levels. Maybe you went on a diet, or maybe you're sleeping and there's no food coming into your body because you're sleeping. So the liver is going to start breaking down triglycerides and glycogen and releasing fatty acids and glucose into the blood. Adipocytes will also break down triglycerides, releasing fatty acids, and skeletal muscles can break down contractile proteins, releasing amino acids. The metabolic reserves are typically stocked when absorption by the digestive tract is greater than the nutrient needs. So the liver will store triglycerides and glycogen. Adipocytes can convert excess fatty acids to triglycerides. The skeletal muscles can build glycogen reserves and use amino acids to increase myofibril numbers, in other words, to grow. So all of these nutrients can either be broken down or rebuilt depending on what the body requires. The diet and nutrient pool do not provide everything that's needed for cellular function. Cellular enzymes are also required to synthesize materials from the available substrates, from the available nutrients. So anabolic and catabolic pathways are coupled. Glycogenesis is synthesizing glycogen from glucose. Glycogenolysis is synthesizing glucose from glycogen. Glyconeogenesis is creating glucose from smaller carbon chains, and glycolysis or glycolysis is the breaking down of glucose into those smaller chains. So here's all of this whole pathway. Triglycerides can be broken down to fatty acids. Fatty acids can be built up into triglycerides. These fatty acids can be broken down into 2 or 3 carbon chains, which can enter the citric acid cycle and electron transport system to produce lots of ATP. Glycogen can be broken down into glucose, or glucose can be built up into glycogen. Either way, it's broken down in times of metabolic need to 2 and 3 carbon fragments to enter the citric acid cycle and electron transport chain. Same thing with proteins. Proteins can be broken down to amino acids, or amino acids can be built back up into proteins, depending on if the body's starving or if the body, has too much. So you can pause the video here, go back and answer these 3 questions. Nutrients are this is the second part of the chapter. So that was the metabolism part. This is the nutrition part. So nutrients are the essential elements in food that are required for normal physiology. Metabolites include things like all nutrients and all the molecules that are synthesized or broken down by those chemical reactions. And digestion is the breakdown physically and chemically of all of those organic compounds. It results in simple compounds that can be absorbed by the digestive tract and distributed throughout the bloodstream. The cells throughout the body are going to use those organic molecules to produce energy and restock that nutrient pool so ATP can be produced. The overall steps in the process of digestion. First, you have the oral cavity, which is where saliva dissolves the organic molecules. Mechanical processing by the teeth and tongue are going to mix everything up with the saliva. Enzymes begin that process of digestion. Then in the stomach, the material is further broken down physically and chemically. Protein digestion begins here. The duodenum, that first segment of the small intestine, is where more buffers from the pancreas and liver are going to moderate the pH, and there's lots of digestive enzymes coming in from the pancreas that are going to catabolize, or that are going to catalyze catabolism. So they're going to bring about more digestive processes. Carbohydrates, lipids, proteins, and nucleic acids are all broken down here. This is also where bile will mix with the chyme at this point. The jejunum is where nutrient absorption occurs. The large intestines where the water is reabsorbed, and the bacteria can produce other nutrients and vitamins, and all of the waste is created and expelled. So carbohydrate digestion specifically begins in the mouth with salivary amylase. Salivary amylase breaks down complex carbohydrates into disaccharides and trisaccharides. It's deactivated as soon as it hits that low pH of the stomach. It is continued in the jejunum and duodenum. The enzymes sucrase, maltase, lactase break down maltose, sucrose, and lactose into simple sugars, and the indigestible carbohydrates provide a nutrient source for bacteria in the colon, and the bacterial activity in the colon can produce gas. So simple sugars from the carbohydrate digestion are going to be transported through the epithelial cells that are lining the jejunum. They diffuse into the blood capillaries, transport to the liver by the hepatic portal vein, and the glucose is typically released into the blood to maintain normal levels around 90 milligrams per deciliter. Insulin allows glucose to go into the cells if blood glucose levels get too high, and when that happens, the liver will convert glucose to glycogen, and the skeletal muscles can do the same thing, use glucose to build up glycogen reserves. Carbohydrates are the preferred substrate for catabolism and ATP production. Proteins or lipids are more important as cellular components, so your cells prefer to use carbohydrates. Glucose is a 6-carbon molecule. It's broken down into 2 pyruvate molecules, which are 3 carbons. This is the process called glycolysis. Look at that word, glycolysis, literally breaking apart glucose. The net gain of that reaction is 2 ATP molecules. This process does not require any oxygen. It's called anaerobic metabolism. It will occur whether or not oxygen is present. The pyruvate is converted into acetyl-CoA, and then it's used in the citric acid cycle. And each pyruvate molecule will yield 34 ATP from the overall metabolic process in the mitochondria. You can pause the video here, go back and review. Let's talk about lipids. Lipid digestion begins in the mouth with salivary lipase. Salivary lipase breaks down triglycerides into monosaccharides and fatty acids. Linolenic and linoleic acid are needed to synthesize plasma membranes. They're called essential fatty acids because they have to come from a dietary source. Our body cannot make them. Lipid digestion continues in the stomach. Chyme in the stomach is going to create large drops of liquids as it mixes around since they're insoluble. The lipase can continue to work but only attack the surface triglycerides at that point. So only 20% of triglycerides are broken down by the time the chyme leaves the stomach. Then well, I kind of skipped this part, but I don't know why it wasn't in there. There are lipase enzymes that are secreted by the pancreas, and so the fat digestion is going to continue from the mixing of bile from the liver and the pancreatic enzymes. Again, not quite sure why there was not a slide on that. Either way, basically the mixing of the lipid or fatty acid with bile will create something called a micelle. A micelle contacts the epithelium and will allow the lipids to diffuse across the membrane. The bile salts can then be reabsorbed and returned back to the liver, and the cells can synthesize new triglycerides from the absorbed lipids. So the lipids come in, and then the cells can repackage them and use them for whatever they need. New triglycerides and cholesterol are basically coated with proteins, and they're called a chylomicron, which is a type of lipoprotein. Chylomicron is water soluble, so is a lipoprotein, so they can float around the blood without needing a transport protein. Chylomicrons are secreted through a process of exocytosis into the lacteals and lymphatic capillaries, and then they will eventually meet up with the venous circulation. This is the whole process of lipid absorption. This is that process here, this step number 3. For some reason, there wasn't a slide on that, but it's right here, and it just has to do with pancreatic enzymes and bile mixing with the lipids to continue their digestion. And they turn into micelles, and micelles will be absorbed. That was the big jump there. So lipid metabolism, once in the lymphatic vessels, the chylomicrons will be brought back to the circulatory system. Fatty acids, monoglycerides are released from the chylomicrons in the capillaries, and the skeletal muscles will use fatty acids for energy, and fat cells will use fatty acids and monoglycerides to make triglycerides, which will be stored. So here's the whole process. This diagram here actually shows you some liver function and cholesterol function. So let's take a look at it. I guess we'll step number 5. I don't know where steps 1, 2, 3, and 4 are. So from the lacteals, I guess they were on this page, 1, 2, 3, and 4. That makes sense. So from the lacteals, the chylomicrons proceed through the lymphatic vessels into the circulatory system. The chylomicrons go into the blood, and then from the capillaries, they will diffuse either into the skeletal muscles to be used for energy, into the adipose to be used to form triglycerides, or they can float back to the liver. Once they're in the liver can absorb the chylomicrons, remove the triglyceride, combine cholesterol from the chylomicron, and alter the surface proteins, and it releases something called a low-density lipoprotein, or an LDL. The LDLs are released by the liver into the bloodstream through the capillaries. Once in the peripheral tissues, the LDLs are reabsorbed by the peripheral tissues. So the cell is going to extract the cholesterol and use it in a variety of ways. Cells need cholesterol, especially in their cell membrane. Then the cholesterol that's not used by the cell is going to diffuse out of the cell membrane and re-enter as an HDL. HDL proteins are released by the liver as well. So the HDLs will return the cholesterol to the liver, where they can be repackaged into a new LDL, or they can just recirculate around as low cholesterol HDLs. So that's the lipoprotein circulation. So here it is in words, if you didn't like my little drawing and my explanation. So from the absorbed chylomicrons, the liver removes the triglycerides, LDLs deliver cholesterol to the peripheral tissues, the cell extracts the cholesterol from the LDLs, putting it back into the blood. HDLs absorb the circulating cholesterol, returns to the liver. In the liver, the cholesterol is then used to synthesize bile and more LDLs, and excess cholesterol is secreted in the bile. Cholesterol is an indication of potential cardiovascular problems. The total cholesterol level should not be above 200 milligrams per deciliter. You should not have a high LDL to HDL ratio. It should be very, very low. So if you do have a cholesterol level above 200 or a very high LDL to HDL ratio, you have potential cardiovascular problems. Values that show high levels of cholesterols would be high HDL. High LDL levels can show the cholesterol that's going to the tissues. Low HDL shows not much cholesterol returning to the liver. So again, if you have high LDL levels, that's saying that there's lots of cholesterol that's going into your tissues, which might not be a good thing. If you have low HDL levels, it shows not much cholesterol is going back to the liver, so it's going somewhere else in your body, which again, might not be a good thing. Excess cholesterol can collect as plaques. So you want to make sure that that excess cholesterol gets back to the liver, which is why you want a high HDL level. You can pause the video here, go back and review. Let's keep on moving through this section. We will move on to protein digestion. The mechanical digestion of proteins begins in the mouth. This is when churning and mixing occurs. There's no chemical digestion of proteins in the mouth. It begins in the stomach for proteins. The strong acidic environment is going to denature the proteins, and then the peptide bonds are exposed to the proteolytic enzyme called pepsin. Pepsin is secreted by the parietal cells as pepsinogen, and then when it's exposed to the HCl, is converted into pepsin. It breaks down proteins into smaller peptide and polypeptide chains, and the digestion of these proteins is completed in the small intestine. Polypeptides and proteins are broken down into dipeptides, tripeptides, and amino acids. Pancreatic enzymes released into the duodenum come from the pancreas. They're secreted as inactive proenzymes, and then they're activated within the duodenum. Enteropeptidase, released by the duodenal epithelium, converts trypsinogen into trypsin, and trypsin will convert other proenzymes to form chymotrypsin, carboxypeptidase, elastase, just other enzymes to break down specific proteins. Peptidases from the small intestine epithelium will break peptide bonds, releasing single amino acids, and those short peptides and amino acids can be absorbed across the apical and basal surfaces of the small intestine epithelial through the process of facilitated diffusion or co-transport. Then they're transported to the hepatic portal vein, to the liver. The liver is going to use the amino acids to make plasma proteins. Some amino acids will be converted into 3 carbon molecules and be used to make glucose. The liver and the body cells can produce 10 of the 20 amino acids that are required. The non-essential ones are the ones that our cells can produce. Essential amino acids are the amino acids that cannot be produced in the body. They have to be obtained from the diet. Meat, eggs, and dairy are the primary sources of these essential amino acids. So here's overall pathway during resting conditions. You're resting here. Carbohydrates, fats, proteins in our food digested into smaller molecules, absorbed throughout the body. We're building this nutrient pool. We can use these nutrients to create lots of ATP, or we can do anabolic pathways and store them for later. You can pause the video here, go back and review these 3 questions. Let's move on to minerals and vitamins. Overall nutrition is the ingestion and metabolism of nutrients from our food. Vitamins are organic molecules that are organic compounds that we need in very small quantities. They typically play a role in our metabolic pathways. Minerals are inorganic ions that are required in very small amounts as well. We have water-soluble vitamins. These include the B vitamins and vitamin C. They're usually components of coenzymes. B vitamins are found in meat, eggs, and dairy. Citrus fruits are a good source of vitamin C as well. They're rapidly exchanged between the digestive tract and blood because they're water-soluble, and they're readily excreted in the urine. Having too much water-soluble vitamin in your body is pretty rare unless you're taking mega doses of multivitamins, like 20 multivitamins a day. Here's a chart with all of the water-soluble vitamins for you, all of the B vitamins. Again, most of them are coenzymes. Do you have to know what all of these vitamins do? No, but you should know what water-soluble vitamins are. Let's talk about how they are absorbed. Most water-soluble vitamins are not stored in the body because they're just going to be excreted in the urine. All but one is absorbed easily in the intestinal epithelium. Vitamin B12 has to bind to something called intrinsic factor. Intrinsic factor is released from the stomach and then travels to the large intestine so that it can help with B12 absorption. Inefficient intake can lead to vitamin deficiency in days or weeks, and intestinal bacteria can produce small amounts of B vitamins, B5, B7, B9, and B12. Bingo. Just kidding. All right, fat-soluble vitamins A, D, E, and K are absorbed through the digestive tract along with lipids, because since they're fat-soluble, they just get sucked in right with the lipids. Sources of these fat-soluble vitamins include vitamins or vegetables. Intestinal bacteria can produce some vitamin K, and the skin can synthesize some vitamin D3 when it's exposed to sunlight. Typically, fat-soluble vitamins are stored in the lipid deposits throughout the body. Normal metabolic activities can continue for months, even if you're not eating them. Hypovitaminosis is a vitamin deficiency disease. Rarely results from dietary insufficiency of fat-soluble vitamins because we can store them. Hypervitaminosis is potentially harmful. This is when the dietary intake exceeds the body's ability to store and utilize or excrete the vitamins. It can be deadly with fat-soluble vitamins. So here's our fat-soluble vitamins. This is the effects of excess, effects of deficiency, and what they do. Do you have to know what the vitamins do? The fat-soluble vitamins, you should be familiar with their source or with their function. Just pick one of the functions and know that for each vitamin. Again, for vitamin C and B, those are just coenzymes. You don't have to know what a coenzyme for. And you should be familiar with the effects of deficiency and excess for these. That's not too overwhelming. Let's talk about inorganic ions like minerals. There are three important roles they play in the body. They can determine the osmotic concentrations of our body fluids like sodium and chloride. They play a role in buffering our body fluids like a variety of various combinations of ions. They're also essential in a variety of enzymatic activities. They act as cofactors with enzymes to catalyze chemical reactions. Sodium, potassium, chloride, calcium, phosphorus, they all have important roles in our body. Do you have to know them? Yeah, you should know their function. You can pause the video here, go back and review this section. Let's talk about proper nutrition. A balanced diet contains all ingredients that's needed to maintain homeostasis. If you have adequate substrates to produce ATP, all your essential amino acids, all your essential fatty acids, vitamins, electrolytes, and water, that would be a balanced diet. It also helps to prevent malnutrition, which is an unhealthy state resulting from inadequate or excessive absorption of one or more nutrients. Your dietary recommendations can be found at choosemyplate.gov, a pretty cool interactive website that you can play around with, where you basically find the balance between the food that you eat and your physical activity level. So it takes into account your age, your weight, your current activity level, and it recommends what your plate should look like every day. And instead of telling you need to eat this many fruits a day and this many vegetables a day, it literally says this is what your plate should look like. It breaks it down a little more, which I think is a little more user-friendly. Energy content in food is expressed in the form of a calorie. Energy needed to rise the temperature of 1 gram of water by 1 degree Celsius is the technical definition of a calorie, but we tend to think of it as something that we don't want to eat too much of because we still want to fit in our clothes. So that's typically what a calorie is. It's an energy term. Carbohydrates yield about 4 calories per gram. Proteins yield about 4 calories per gram. Lipids yield about 9 calories per gram. Alcohol is a kind of odd category. It's not listed here, but it yields 7 calories per gram. So it's very high energy content in alcohol, hence beer bellies and that type of a thing. The average adult needs 2,000 to 3,000 calories a day. I would say that's a little high. The average adult needs probably between 1,600 and 2,500 calories a day, depending on activity level. Just looking at some overall foods and their calorie ranges. Most carbohydrate foods are high in calories. Most protein foods are low in calories. And obviously fatty foods have high calorie content as well. There are 5 basic food groups and they have differing effects on health: grains, vegetables, fruit, dairy, and meat. You should be familiar with them. You're probably already familiar with them if you've ever taken any type of health or biology class before. These are typically covered. So let's get into some more complex stuff that's probably new to you, the idea of a complete or an incomplete protein. A complete protein is a protein that provides all of the amino acids in sufficient quantities. These are our animal products. Dairy, beans, well, beans obviously are animal products, meat, eggs, those provide sources of complete proteins. Incomplete proteins contain inadequate amounts of amino acids. They lack the essential amino acids. So these are typically vegetable products, but you can combine different vegetables to still create complete proteins, which is why being a vegetarian can still be a healthy option. There's complete proteins for you. Incomplete proteins come from plant sources, but again, combining and eating enough plant proteins will create complete proteins for your diet. You can pause the video here, go back and review this section. Let's talk about how our appetite is controlled. There is a feeding center or satiety center, which is within our hypothalamus. It's involved in the controlling our appetite. It has opposite effects. It is affected by multiple factors. There's social, psychological factors, dietary habits, etc. For example, when I'm done lecturing, don't do what I do. I have my chips and my snack here because I get awfully hungry talking for about an hour. I have my water here. This is my habit. This is what I do. I decide I'm going to come down here in my office, shut the door, snack a little bit, and then I'm going to give a lecture, and then I'm going to snack some more. That's what I do. Or maybe you know that when you get together with your girlfriends on a Friday night, you're going to drink margaritas and, you know, eat, I don't know, cheesecake or whatever. That's just what you do. That's like a societal pressure. Or every holiday, this is what you eat. Or on your birthday, you eat cake, right? Those are things. There's also psychological pressures that you feel, like when you're around, you know, at the football tailgate and everybody's eating the buffalo chicken dip and you're trying to be good, but you really want some. So there's other pressures like that, as well as just various habits, like getting in the habit of eating breakfast every day type of a thing. So they're all unknown mechanisms and pathways to our appetite control, but there's also some very specific physiological factors that control our appetite. It involves glucose levels and hormones. There's neuropeptide Y, NPY, and ghrelin. They stimulate the feeding center in our brain, and leptin stimulates our satiety center. So neuropeptide Y and ghrelin are acting opposite of leptin. So here's the overall appetite regulation pathway. You have short-term regulation and a long-term regulation. The short-term regulation through the satiety centers, elevated blood glucose levels are going to depress the appetite. Low blood glucose levels are going to stimulate the appetite. Hormones in the digestive tract, like CCK, suppress after a meal, and stimulation of stretch receptors in the digestive tract are going to suppress the appetite as well, cause that sense of feeling full. Our feeding center is going to be activated when our neuropeptide Y, for example, is stimulating the feeding center to produce that appetite. Or ghrelin, when our stomach is empty and the stomach is basically sitting around saying, I'm empty, I need something else in me. So ghrelin will be released and causing you to be hungry. The long-term regulation is when the appetite outpaces energy usage. So excess calories are stored as fat in the adipose tissue. Adipose tissues release the triglycerides and they release a peptide hormone called leptin. In the central nervous system, leptin is going to stimulate the satiety center and suppress the appetite. So it's going to make you feel full as soon as lipogenesis happens, as soon as those fat cells start to accumulate. Usually that is after you're already full, and then you say, oh, I really ate way too much. I need to unbutton the pants. There's also eating disorders. Unfortunately, these are psychological problems that result in inadequate or excessive food consumption, and there's many different forms. They all have an obsessive concern with food and body weight. Anorexia is the lack or loss of appetite. Anorexia nervosa is a form of self-induced starvation. It's most common in Caucasian females. Death rates can be 10 to 15%. Bulimia is where the individual binges, eating up to 20,000 calories and then induces vomiting, or laxative use can be used as well, diuretics as well. It's more common than anorexia nervosa, and again, generally affects adolescent females. There's obesity, which is defined as being 20% over your ideal weight. It's linked with serious health risks. Your body mass index can be calculated by dividing your weight by your height and the desire. And there's tons of, if you've never calculated your BMI, just Google BMI calculator and type in your BMI, and it will even tell you if you're within these particular numbers. So if you're within the range of 19 to 25 after you divide those two numbers, then you're in a very healthy weight. If you're overweight, then you're 25 or over, and if you have a BMI of 30 or more, then you're considered obese. Obese. If you're under 19, then you're underweight. That isn't listed on here. There are several types, or there's two types of obesity. Regulatory obesity is the most common form. This is just the failure to regulate food intake. In other words, you eat too much. And there's also metabolic obesity, which is relatively rare. And there's usually some type of metabolic malfunction causing that. This guy is surprised by how much he weighs. You can pause the video here, go back and answer these three questions. Some metabolic disorders which result from inadequate nutrition or a biochemical problem or starvation. PKU, protein deficiency diseases, ketoacidosis, and gout are some of them. Let's talk about phenylketonuria, or PKU. This is an inherited metabolic disorder. This is the inability to produce a specific enzyme needed for amino acid metabolism. You cannot convert phenylalanine into tyrosine. So as a result, they can't synthesize epinephrine or norepinephrine or dopamine or melanin. If it's not detected at birth, the brain damage will result. Treatment is very low, phenylalanine diet, and a protein supplementation. Typically, newborns are tested for PKU as soon as they're born. They get a vitamin K shot to help with blood clotting, and they get a PKU test right away just to make sure. Because if it's caught early on, it's fine. If it's not caught on early on, then again, severe brain damage can occur. So protein deficiency diseases are decreased protein synthesis from insufficient uptake of essential amino acids. So plasma proteins can be broken down faster than the liver can replace them, ultimately reducing the plasma osmolarity, fluid shifting in and out of the capillaries. This could result in ascites, accumulation of fluid in the peritoneal cavity, or edema, which is the accumulation of fluid in the interstitial spaces. Kwashiorkor, or protein deficiency disease in children, is when the protein intake is deficient enough. Even if sufficient total calorie intake. So complications include damage to the developing brain. This is the puffy belly on the starving children. Very sad. Kwashiorkor. Ketoacidosis, if limited glucose supplies and the liver breaks down fatty acids and amino acids for energy, basically this is going to elevate your acetyl-CoA levels, producing lots of ketone bodies. These ketone bodies will accumulate and lower the blood pH, causing ketoacidosis. The blood pH can drop below 7.05, causing a coma, cardiac arrhythmias, and even death. So uncontrolled diabetes mellitus is a risk for this. If there's no glucose available for the liver is going to respond as if it's starving by breaking down protein and producing these ketone bodies. Gout is when RNA is recycled. The purine bases, adenine and guanine, are catabolized into uric acid. It's categorized as a nitrogenous waste. The normal uric acid concentration is 2.7 to 7.4 milligrams per deciliter, but over 7.4 milligrams per deciliter, it's going to start saturating our body fluids, and it becomes insoluble crystals, and that's what we call gout. It typically accumulates in the joints, usually in the big toe. Gouty arthritis can persist for several days and then disappear and then come back. Patients are just advised to limit meat, beer, and seafood consumption. It's just painful. Obviously, this person needs pedicure. So do I. But this is a very painful condition. As you can imagine, little tiny crystals building up in the joint cannot be comfortable. So starvation and metabolic reserves. The metabolic reserves are the organic substrates that are available to the body for catabolism. Lipids, proteins, carbohydrates. The adipose tissue has very high concentrations of triglyceride reserves. Protein reserves are within the contractile proteins of our skeletal muscle, and there's very little carbohydrate reserves in our body, just enough for a few hours. So with prolonged starvation, the body's going to start to catabolize carbohydrates first, then fatty acids, then proteins. So here you see the overall breakdown. Nutrients are stored. They're broken down by glucocorticoids in the liver. Lipids and amino acids break down when they're stimulated by glucocorticoids in the process of gluconeogenesis or glycogenolysis. They're going to create ketone bodies or glucose. Ketone bodies are used for ATP production, stimulated by glucocorticoids. Glucose is used for ATP production in the neural tissue. The adipose tissue is broken down into lipids, used for ATP. Skeletal muscles can break down proteins or do glycolysis, again, to produce amino acids or lactate. Lactate is going to be used for gluconeogenesis to make glucose, which is used for ATP. And amino acids are, again, used for the same, gluconeogenesis. So this chart here showing you changes in the metabolic stores over time in weeks. So you have the carbohydrate line, you have the fatty acid line, and then you have protein line. So this is showing you how our energy reserves are declining over weeks. Notice the very sharp decline in carbohydrates, followed by a thinner line of lipids, fatty acids, and then proteins don't decline up until after about 6 weeks. So, I mean, they do decline, but not dramatically, again, because your proteins are your muscles, your skin, your hair, your nails. So that's basically what your body is. So the proteins are going to be the last thing to be used. You can pause the video here, go back and review. Let's talk about the final little chapter, which is metabolism and body temperature. Nutrition and metabolism affect our body temperature. Foods consumed are a mix of carbohydrates, fats, and proteins. Organic molecules release small sugars, fatty acids, amino acids are transported and absorbed through the body. The cells absorb the nutrients, use them to generate ATP. Remember, ATP is just 40% of the energy from the food. 60% is heat, right? So that heat is warming the cell, warming the surrounding tissue. The metabolic heat can be produced from a variety of activities, and our body temperature is maintained by homeostatic mechanisms. So this is all done through the study of energetics. This is the study of the flow of energy as it changes from one form to another. The amount of energy required for activities varies from moment to moment. So we calculate something called the BMR, our basal metabolic rate. This is the minimum resting energy expenditure for someone who is awake and alert. If you were just sitting in your chair all day, not doing anything, this is how much energy you would burn. It's using something called energetics. It can measure, you can measure it directly by measuring your respiration rate, or you can calculate it. This is obviously taking a very complex computer system, although it does look like kind of 1980s picture, but taking an actual BMR is pretty complicated. You can do the same thing you did with BMI. Just Google BMR calculator, you'll calculate your basal metabolic rate. The average individual has a BMR of around 70 calories per hour. Varies with your size, your gender, your weight, and how much physical activity you partake in on a daily basis. But your food intake has to equal that number. Or else you're going to be either undernourished or overnourished. So the estimated calories expended by a 70 kilogram, or what is that, like a 170 pound person. So speed walking for an hour, you're going to burn about 400 or so calories. Competitive swimming, you're going to burn an awful lot of calories. That's something we probably know, right? You exercise, you burn calories. Makes sense, right? Thermoregulation are the activities that generate ATP, as we mentioned, also generate heat. There's enzymes that are needed to get that optimal temperature, and it's through the process of thermoregulation. The lack of control of thermoregulation can lead to death and dysfunction. The body transfers heat through the process of radiation, evaporation, convection, and conduction. Radiation is more than 50% of the heat loss indoors is from radiation, just from radiating off of us. Evaporation is when water changes from liquid to a vapor, and the sweat glands do this for us. It evaporates off of our body, cooling us down. Convection is when heat is lost over the surface of the body. The warm air rises away from the body and it's replaced with cooler air. This is about 15% of our heat loss. And conduction is the direct transfer of energy. Typically, we don't conduct. I guess if I were, thinking maybe my son, if he got out of the pool and he was very cold and I took my arms and I like rubbed it on him and took my body heat and, put my hands on his back or something like that, the heat from my hands would warm him up. That's conduction. So direct contact is conduction. So here you see some underlying physical conditions related to our body temperature. Our body temperature likes to be in this normal range, but if you get up in this 106 or above range, that's severe central nervous system damage, death, convulsions, etc. Same thing with below, even just below 90 degrees can be very detrimental, loss of muscle control, etc. You can pause the video here, go back and answer these 3 questions. We're winding things down. There's something called the heat loss center. The heat loss center is going to trigger the loss of heat from your body. So the behavioral changes like getting to the shade or drinking water, vasodilation and shunting blood to the skin surface to allow that heat to escape from the body, increased sweat production, respiratory heat loss, and increasing deeper breaths and increasing our rate of respiration to get rid of that hot air that's inside of us. This is all coordinated through the hypothalamus. Then we have the heat gain center. This is when the falling body temperature triggers that response, increasing the generation of heat as well as the conservation of heat. So you have shivering and non-shivering thermogenesis. Non-shivering thermogenesis is when hormones raise your body metabolic level. Shivering thermogenesis is when you're actually shivering and that contraction creates body heat. Overall conservation of body heat is done through the vasomotor center, decreasing blood flow to the dermis, reducing heat loss through radiation and convection. And then there's countercurrent exchange, which is where the heat is transferred from deep arteries to adjacent deep veins, and it traps heat close to the body core and reduces heat loss. So this is why you have those superficial veins and deep veins in your arm, again, just to help with that regulatory process. Last slide here, you can stop the video, answer these 3 review questions. Please let me know what questions you have about Chapter 16, and have a great night or day, whatever time of day it is for you. Bye-bye.